Welcome to the deep dive. Our mission here is pretty simple. We take complex, fundamental source material, we cut through all the noise, and we deliver the essential insights you need.
And today, we are going deep on what is, you could argue it's the most perfectly engineered protein in existence.
Hemoglobin.
Hemoglobin. You really can't overstate it. I think you have to start with the evolutionary context to really get it.
Right.
Life began, mostly anaerobic. It survived without free oxygen. And then this huge shift happened, the ability to actually use oxygen.
The transition to aerobic respiration. It was a complete energy revolution.
It absolutely was. We're talking about extracting something like 15 times as much energy from a single glucose molecule if you have oxygen around compared to just, fermentation.
15 times the power. That is a game changer for biology.
A total game changer. That energy upgrade is what allowed organisms to become so much bigger, so much more complex. But, that complexity created its own problem.
A logistics problem.
Exactly. Simple diffusion of oxygen just, it wasn't going to cut it anymore. You can't just rely on things passively spreading when you've got trillions of cells buried deep inside tissues.
So the body needed a specialized supply chain. It needed a way to deliver this powerful new fuel oxygen from, the outside world to every single cell.
Right. And for vertebrates, that meant you needed two essential components. First, a robust circulatory system, and second, these specialized carrier proteins.
And those are the proteins we're focusing on today.
Hemoglobin, the great transporter tucked inside your red blood cells, and its evolutionary cousin, myoglobin, which you find mostly in your muscle tissue.
So if hemoglobin is the sophisticated, regulated shipping company, is myoglobin kind of the local, high-capacity warehouse?
That's a perfect analogy. Hemoglobin is built for efficient, regulated transport. It picks up oxygen in the lungs, dumps it in the tissues. It's so efficient, it uses up to 90% of its potential capacity in just one trip. Yeah. Myoglobin, on the other hand, it has an incredibly high affinity for oxygen. It just grabs it and holds on, which makes it a fantastic storage unit. It helps oxygen diffuse from the blood into the muscle cell, and, it's the reserve supply for when demand gets really high.
What's just amazing to me is that these two related molecules, which share the same core structural blueprint, do these completely different jobs.
And that difference, it comes down almost entirely to how the four subunits of hemoglobin are put together, its quaternary structure.
So our goal today is to really use the supplied biochemical research to dissect that relationship, how structure dictates function, and how these tiny molecular shifts create profound physiological control.
We're going to get into the stunning molecular details. We want to understand how a shift of less than half an angstrom, a movement of a single iron atom, is basically the mechanical trigger that dictates whether you live or die.
And how the protein responds to its environment, things like acidity and waste products.
Right. And you can't skip the history here either. These proteins were, pioneers. Hemoglobin and myoglobin were the very first proteins to have their complex three-dimensional structures solved.
That was using X-ray crystallography by Max Perutz and John Kendrew back in the 50s.
That work was foundational. Until then, proteins were just sort of unknown blobs. Seeing that globin fold for the first time completely changed our understanding of structural biology.
And it was hemoglobin that gave us the first molecular proof that a single amino acid change could cause a disease.
Linus Pauling's work on sickle-cell anemia. Yeah.
Yeah.
We are really standing on the bedrock of modern molecular biology here.
Okay, so let's start with that basic blueprint, the single subunit molecule, myoglobin. What does its structure tell us about its role as that local oxygen store?
Myoglobin is the simplest starting point. It's a single polypeptide chain, and its structure is made up mostly of alpha helices linked together by turns. They all wrap up into this compact globular shape that we call the globin fold.
And it can exist in two states, right? Deoxy or oxy.
Deoxyhemoglobin, which is oxygen-free, or oxyhemoglobin, with oxygen bound. And that structure, the one Kendrew determined, was really the first time we could visualize a protein's 3D architecture. It was a huge moment.
But the protein chain itself, it doesn't actually bind the oxygen directly. It needs an extra piece of equipment to do the job.
A prosthetic group. That's the crucial heme molecule. It's bound very tightly. It's absolutely essential, and it's what gives blood and red muscle their characteristic color.
And the heme group itself is complex. It's got this big organic ring structure.
Protoporphyrin.
Right, protoporphyrin with an iron atom sitting right in the middle.
When you look at it, it really does look immense. It's made of 4 smaller rings, these pyrrole rings, all linked together. You could almost picture it like a big clover leaf.
So what's the purpose of this huge organic structure?
Well, its job is to create a very specific chemical environment for that central iron atom and the iron. The iron is the true star of the show.
Because for oxygen binding to even happen, that iron has to be in the right state,
The ferrous, or Fe2 plus oxidation state. If it gets oxidized to the ferric, E3 plus state, you get what's called myoglobin.
which can't bind oxygen at all.
Not at all. So you have to maintain strict chemical control over that iron. It's paramount.
Okay, so the iron atom sits in the center, and it has 6 potential binding points, or coordination sites. 4 of them are taken up by the nitrogen atoms of that big ring. How does the protein chain latch on?
The protein uses the 5th coordination site. There's a residue called the proximal histidine, and it extends its imidazole ring to bond directly with the iron.
So that's the tether. It anchors the heme inside the globin pocket.
Exactly. And the 6th coordination site, that's the open one. It faces out into the binding pocket, and that is where the oxygen binds.
Now we get to the really pivotal moment, the molecular mechanism of action. The source material puts a huge emphasis on this tiny yet massive structural shift when oxygen binds.
Right, this is the cause and effect relationship that's driven by that iron atom.
So what happens?
In the deoxyhemoglobin state, so without oxygen, that ferrous ion is actually a little bit too large to fit perfectly flat inside the porphyrin ring.
So it's puckered.
Yeah, it sits just slightly domed, about 0.4 angstroms outside the plane of the ring, getting pulled toward that proximal histidine.
And an angstrom is 10 to the minus 10 meters. So
Yeah.
0.4 angstroms is a minuscule distance. It's far smaller than the diameter of a hydrogen atom.
It's minuscule, yet it is the entire mechanical key. Because when oxygen binds to that 6th site, it completely changes the electronic structure of the iron.
And that rearrangement makes the iron ion effectively smaller.
Right. And crucially, that tension from the proximal histidine is overcome, and the iron atom snaps into the plane of the porphyrin ring. It's rapid, it's reversible, and it defines the change between the 2 states of the protein.
And this tiny structural shift has real-world consequences, even beyond biology. That change in electronic structure is what's leveraged by modern medical imaging.
Absolutely. This is the basis of functional magnetic resonance imaging, or fMRI.
How does it work?
Well, deoxyhemoglobin, with its displaced iron, has magnetic properties that are distinct from oxyhemoglobin, where the iron is sitting nicely in the plane. fMRI can non-invasively detect those differences.
So if a part of your brain becomes really active, it needs more oxygen, so blood flow increases there.
Right. And that increase shifts the magnetic profile that the scanner detects.
Ah.
You're watching it go from a high proportion of deoxyhemoglobin to a high proportion of oxyhemoglobin in real time.
So when we see an fMRI scan showing which parts of the brain light up, we're basically just watching the real-time movement of iron atoms inside the heme groups of the local blood supply.
That's it. It's an astonishing connection between basic biochemistry and high-tech diagnostics.
Wow.
But let's go back to that binding pocket. Once oxygen is bound, the job's not over. You have to make sure it can be released safely and, reversibly.
And the binding itself, it's not a simple attachment. It involves some electron transfer.
A partial electron transfer. It's best described as a complex between the ferric ion F3+ and a superoxide anion, O2-.
Which sounds dangerous. Superoxide is a highly reactive oxygen species. It's a classic damaging radical.
It is. If the protein released superoxide, it would be losing its functional capacity permanently.
Right, because the critical constraint is that the protein has to release O2 dioxygen, not O2-, the superoxide.
Exactly. Releasing superoxide would leave the iron permanently oxidized to the ferric state, F3+, and you'd have myoglobin, which, as we said, loses its oxygen binding ability.
So how does the protein prevent this catastrophic side reaction? How does it maintain the reversibility?
This is where the distal histidine comes in.
Okay, so we had the proximal histidine tethering the iron.
Right. The distal histidine is on the other side. It's located just slightly above the binding site, positioned perfectly to form a hydrogen bond with the bound oxygen molecule.
This hydrogen bond seems like a simple feature, but what's it actually doing?
That hydrogen bond stabilizes the bound oxygen. It specifically strengthens its O2 character and makes it much less likely to be released as the superoxide radical. The protein structure is basically a chemical gatekeeper, controlling the heme's own reactivity to make sure the binding is reversible.
And it does something else too, right?
Yes, it plays a second critical protective role. It sterically hinders the access of carbon monoxide, CO,
The poison.
the poison. CO prefers to bind to iron in a perfectly straight, linear line, but the bulky distal histidine forces it into a less stable, angled binding configuration.
So it slightly reduces its affinity compared to what it would be in an unprotected environment.
Exactly.
Okay, so myoglobin is the single unit structure, high affinity, great for storage, engineered for stability and safety. Now let's scale up. Let's look at hemoglobin, the transport unit.
Human hemoglobin A, or HbA, is a tetramer. That just means it's made of 4 polypeptide chains.
2 identical alpha chains and 2 identical beta chains.
Right. And crucially, every single one of those 4 subunits still contains a heme group and structurally shares that same underlying alpha helical architecture, the globin fold that we saw in myoglobin.
That shared fold is the clear footprint of their common evolutionary origin.
It is. And the way these 4 pieces are assembled is key. Hemoglobin functions as a tightly associated pair of identical alpha-beta dimers.
So we call them alpha-1 beta-1 and alpha-2 beta-2.
And the association of those 2 dimers forms the full tetramer. The interface between them is extensive and highly regulated. And one last important point. The 4 heme groups are actually quite far apart.
How far?
Oh, typically separated by 24 to 40 angstroms. And that physical separation is vital because it proves that binding at one site must somehow be transmitted mechanically to influence the affinity of the others.
Which leads us directly into cooperativity.
Exactly.
This is where we see that dramatic functional separation.
Yeah.
Myoglobin has this high affinity, a simple, predictable binding curve. But hemoglobin has to be a carrier.
Which means it has to efficiently bind oxygen in the lungs and then efficiently release it in the tissues.
How do the binding curves show this difference so clearly?
For myoglobin, the curve is a simple, high-affinity hyperbolic curve. It hits half saturation, what we call P50, at only 2 torr partial pressure.
So that protein is basically always saturated under most physiological conditions.
Right, which makes it a terrible transporter. It would just hold the oxygen hostage.
And hemoglobin?
Hemoglobin's curve is totally different. It's S-shaped, or sigmoid. And that shape instantly tells us 2 things. First, its affinity is much lower overall. Its physiological P50 is about 26 torr in red cells.
And second, and this is the important part,
The sigmoid shape is the unmistakable signature of cooperative binding.
explain what that means in practical terms.
Cooperative binding just means that the binding of the first oxygen molecule to one subunit in the tetramer increases the likelihood, the affinity of the remaining 3 sites binding oxygen.
The subunits are communicating with each other.
They're communicating. And the reverse is also true. When the first oxygen molecule is released in the tissue, it signals the other 3 sites to let go of their oxygen as well. This makes the whole delivery process incredibly efficient.
Let's use some numbers here because this is where the genius of the design really becomes clear. How much more effective is cooperative binding for transport?
Well, think about the physiological context. In your lungs, the partial pressure of oxygen, the PO2, is high, around 100 torr. At that pressure, hemoglobin is about 98% saturated.
Okay, it's fully loaded.
Fully loaded, then that blood gets to your metabolizing tissues, and the PO2 drops significantly, maybe down to 20 torr.
And what happens to hemoglobin saturation?
It plummets to 32%, so the amount delivered is just the difference. 98 minus 32, which is 66% of its potential oxygen capacity.
So 2/3 of the total load is delivered in just one circuit. That is impressive efficiency.
It is. Now, for contrast, imagine if myoglobin were the carrier.
Okay.
It would still be 98% saturated in the lungs, but in the tissues at 20 torr, it would remain stubbornly 91% saturated.
So it would only release 7% of its load.
Only 7%. Cooperativity allows hemoglobin to deliver nearly 10 times the amount of oxygen that myoglobin would, and almost twice as much as any theoretical non-cooperative protein. The sigmoid curve is just essential for effective transport.
And it's tuned perfectly. The steepest part of that S-curve is right between 40 torr, which is typical for resting tissues, and 20 torr for exercising tissues.
Right. And in that specific range, a relatively small drop in oxygen concentration results in a huge release of oxygen. Nearly 45% of the total load gets delivered just in that range.
So it ensures that oxygen delivery is maximal precisely when and where the tissues need it most.
Exactly.
So we know the subunits communicate, but how? The heme groups are physically separated by tens of angstroms, so this has to be a mechanical process, a quaternary structural shift.
Precisely. Hemoglobin operates between 2 main quaternary structures. The deoxygenated form is called the T state.
T for tense.
Tense. It's stabilized by tons of salt bridges and subunit interfaces, which puts a strain on the heme pockets and gives them a low oxygen affinity.
And the oxygenated form.
That's the R state for relaxed. Oxygen binding breaks those constraints. It relaxes the structure, and that leads to a much higher oxygen affinity.
And the physical shift we see is the rotation of those alpha-beta dimers relative to each other.
Yes. The alpha-1 beta-1 and alpha-2 beta-2 dimers undergo this coordinated relative rotation of about 15 degrees. That rotation is what snaps the protein from the constrained T configuration into the more open R configuration.
Let's trace the molecular mechanism. How does binding a single oxygen molecule translate into that massive 15-degree rotation across the whole complex?
Okay, this brings us right back to that 0.4 angstrom shift.
The iron moving into the porphyrin plane.
Right. When O2 binds, the iron moves. And because the iron is physically tethered to the protein by that proximal histidine at the 5th coordination site, the iron's movement pulls the proximal histidine with it.
So the iron atom is like the trigger. The proximal histidine is the lever.
That's a great way to put it. That histidine residue is part of a specific alpha helix within the subunit. So when the histidine is pulled, the entire helix has to move with it.
And the end of that moving helix lies right at the critical interface between the alpha-beta dimers.
Exactly. So that tiny local movement is amplified by the mechanical lever of the helix, and it forces a change in the subunit interactions, which kicks off the T to R transition and that 15-degree rotation of the entire tetramer.
It's a nanoscale machine, really, designed to transmit information through strength. Now, this whole idea of subunit communication led to different theoretical models for cooperativity.
Right, the concerted versus the sequential models.
They're idealized tools for understanding these allosteric proteins. What's the concerted model?
The concerted model, or MWC model, it assumes that tetramer exists in only 2 states, T or R. All 4 subunits have to transition together instantaneously.
So oxygen doesn't induce the R state, it just shifts the equilibrium toward it.
Precisely. It shifts the overall population equilibrium towards the R state simply because the R state has a higher affinity.
Okay, and the sequential model?
The sequential model is different. It says that ligand binding to one site changes only that one subunit's conformation. Then that local change induces conformational changes in the neighboring subunits, increasing their affinity one by one.
Sequentially.
Right, step by step, without necessarily causing a full global T to R flip right away.
And where does real-life hemoglobin fit? Is it a combination?
It is a combination. It leans very heavily toward the concerted model. For instance, once 3 oxygen molecules are bound, the quaternary structure is almost always the R state.
But there's a sequential element too.
There is. Hemoglobin with only 1 oxygen bound is still structurally in the T state, but it already binds the 2nd oxygen molecule 3 times more strongly than a fully deoxyhemoglobin.
So that intermediate affinity increase suggests a local change has already been transmitted, which is a key feature of the sequential model. This brings us to a really critical internal regulator, 200-3-bisphosphoglycerate, or 213-BPG. If we purify hemoglobin in the lab, it binds oxygen way too tightly.
Yeah, it acts almost like myoglobin. It would only release about 8% of its cargo. It would be a terrible transporter.
So what's wrong with purified hemoglobin, and how does 213-BPG fix this problem?
The problem is that purified hemoglobin, lacking all the stabilizing factors you find in a red blood cell, is inherently unstable in the T state. It strongly favors the R state.
And the solution is 213-BPG.
The solution is 213-BPG. Red blood cells maintain a remarkably high concentration, around 2 millimolar, of this highly anionic compound.
So where does this highly charged molecule bind, and how does it stabilize the T state?
It binds right in the central cavity of the hemoglobin tetramer. This pocket is formed exclusively by the beta chains, and importantly, it is only available in the T or deoxy state.
And the molecule is highly anionic. It has 5 negative charges.
Which is perfect for forming extensive salt bridges with all the positively charged residues that line that cavity.
Which residues specifically are involved in locking it in place?
On each of the 2 beta chains, 2003-BPG interacts with the side chains of lysine 82, histidine 143, and the N-terminus of the beta chain itself, plus a few other sites.
So a single molecule of 2000-BPG forms 7 strong ionic bonds. It's like a clamp.
It's like a clamp. It locks the 2 beta chains together, stabilizing the T state.
So 2003-BPG is the energetic cost required to keep hemoglobin in the low-affinity T state until oxygen forces the shift. To switch to the R state, that central pocket has to collapse, and the 2003-BPG has to be expelled.
That's it. By stabilizing the T state, 2003-BPG forces more oxygen to bind before the energetic barrier for that T to R shift is overcome. And that's what lowers the overall oxygen affinity to the physiological P50 of 26 store, which makes transport efficient.
This is the definition of allosteric regulation.
A regulator binding to a site separate from the active site to modulate activity. Yep.
And this mechanism explains that crucial difference between adult and fetal oxygen handling. Fetal hemoglobin needs to have a higher affinity than the mother's adult hemoglobin.
That's right. It has to be able to steal oxygen from the mother's blood across the placenta.
So it's made of alpha-2 gamma-2 chains instead of alpha-2 beta-2.
And that fetal gamma chain is very similar to the adult beta chain, except for one critical substitution right in that 2-3 BPG binding pocket. A neutral serine residue replaces the positively charged histidine 143.
So by removing that histidine, you're removing two positive charges from the fetal hemoglobin's 2x3 BPG pocket.
Which substantially reduces its ability to bind the negatively charged 2-valery BPG. As a result, fetal hemoglobin is less stabilized in the low-affinity T state and spends more time in the high-affinity R state.
Which allows it to efficiently pull oxygen across the placenta. It's a brilliant example of specialized gene expression solving a major physiological problem.
It really is.
Before we move on, let's just briefly circle back to carbon monoxides and CO. We mentioned that distal histidine helps protect us a bit, but CO is still devastating because it's a double-edged sword.
It is the silent killer. It forms carboxyhemoglobin by binding to the exact same site on the heme iron as oxygen does, but its affinity is roughly 200 times higher.
So even tiny amounts of CO in the air can completely incapacitate a huge amount of your hemoglobin capacity.
That's the first problem, the physical blockade. But the second effect is just as fatal.
What is it?
The second effect is that CO acts as a super allosteric effector. When CO binds to just one subunit, it forces the entire tetramer into the high affinity R state.
So it dramatically increases the affinity of the remaining three unoccupied sites for any available oxygen.
Right. The hemoglobin essentially won't let go of the oxygen it manages to pick up. It starves the tissues. Your blood might look bright red, but it's biologically useless for delivery.
Which is why the treatment is high-pressure, pure oxygen.
Yeah.
You have to radically increase the PO2 in the lungs to just win that competitive binding battle and physically displace the CO.
That's the only way.
This is where we see hemoglobin truly function as a logistics expert, an environmental sensor. When tissues work hard, they generate waste. Hydrogen ions, which means a drop in pH, and carbon dioxide.
And hemoglobin has evolved to sense these cues and respond by dumping more oxygen. This is the Bohr effect.
A magnificent piece of self-regulation.
It is. Like 2,3 BPG, hydrogen ions, and CO2 are allosteric effectors. The high levels of metabolic waste products signal high demand, and those same waste products trigger the release of the required oxygen.
Let's focus first on the hydrogen ions, or pH. We feel that pH drop when our muscles start to burn during strenuous exercise. How significant is that affinity decrease?
It's very significant. Let's say blood moves from the lungs, where the pH is optimal at 7.4, to active muscle, where it's more acidic, maybe pH 7.2.
Okay.
If the pH stayed at 7.4, about 66% of the oxygen would be released. But because the pH drops to 7.2, the oxygen release jumps to 77%.
So that's an 11% boost in delivery capacity.
Triggered purely by that slight increase in acidity.
How does the protein mechanically sense a slight change in the concentration of protons?
Well, hemoglobin has several ionizable groups whose pKa values are carefully positioned right around the physiological pH of 7.4. They act like molecular pH switches.
And the most critical example is a specific histidine residue.
Histidine beta 146, located right at the carboxyl terminus of the beta chain. Histidine residues are often crucial for this because their side chains can easily flip between being charged or neutral in this pH range.
So what happens when the pH drops?
In a more acidic environment with more hydrogen ions around, his beta 146 readily accepts a proton and becomes positively charged.
And then what?
In the deoxyhemoglobin, the T state, this newly charged histidine residue snaps into position to form a critical salt bridge, an ionic bond, with a negatively charged neighboring residue, aspartate beta 94.
So low pH leads to protonation of his beta 146, which forms a salt bridge with as beta 94, which stabilizes the T state.
And that stabilization is the whole point. By reinforcing the T state, oxygen release is favored. Then, when the blood returns to the lungs, the high PO2 drives the T to R transition, which physically breaks these salt bridges, causing his beta 146 to release its protons.
So the protein is literally absorbing protons in the tissues and releasing them in the lungs.
It is. Which brings us to the second effector, CO2.
Right, the other major metabolic waste product. And it stimulates oxygen release in two different ways. What's the main way?
The primary mechanism is indirect, through the pH drop we just covered. CO2 rapidly passes into the red blood cell, where it finds this incredibly fast enzyme called carbonic anhydrase,
And that enzyme catalyzes the reaction of CO2 with water to form carbonic acid.
H2CO3. And carbonic acid is unstable. It dissociates instantly into bicarbonate, HCO3-, and crucially, a free hydrogen ion, H+.
And that burst of H plus drops the local pH inside the red cell.
Which, as we just established, triggers the Bohr effect and stabilizes the T state via his beta-146 salt bridge. So the bulk of CO2's regulatory effect is through acidification.
So if that's the indirect mechanism, what is the direct chemical interaction between CO2 and hemoglobin?
CO2 can also react directly with the uncharged terminal amino groups of the hemoglobin subunits.
The NH-NH2 group.
Right, and it forms these negatively charged carbamate groups, RNHCO-.
So a carbamate group just adds a negative charge to the end of the hemoglobin chain.
Precisely. And this new negative charge is perfectly situated to participate in new salt bridge interactions that further stabilize the T state, completely independent of the pH drop.
So CO2 has a 1-2 punch. It acidifies the environment, and it directly modifies the protein to lock it in the low affinity state.
It does. It ensures maximum oxygen delivery. When blood leaves the tissues, the combination of H+ and CO2 ensures that oxygen release is approaching 90% of maximum capacity under those local conditions.
And this carbamate formation, it's not just regulatory, it's also functional for waste transport.
Right. About 14% of the CO2 generated in the tissues is transported back to the lungs, bound directly to hemoglobin as carbamate.
So what about the other 86%? How is the bulk of the CO2 moved?
Most of it is transported as bicarbonate, HCO3-, dissolved in the blood plasma. The bicarbonate is formed inside the red blood cell by carbonic anhydrase. Then it leaves the red cell and enters the plasma through a special membrane transporter.
The anion exchanger, or chloride shift protein.
Yeah, it exchanges the outbound bicarbonate for an inbound chloride ion to maintain electrical neutrality.
And this entire process just reverses in the lungs.
It reverses completely. High oxygen in the lungs forces the T to R shift, which breaks the salt bridges and forces the release of H+. That increase in pH favors the reversal of the carbonic anhydrase reaction.
So H plus recombines with bicarbonate to form CO2 and water, and the CO2 is exhaled.
It's a beautifully coordinated mechanism of transport, delivery, and waste removal.
We've spent all this time emphasizing how perfectly tuned hemoglobin is. Now we have to look at what happens when a single atom or a single amino acid is changed. This brings us to sickle-cell anemia.
The disease that first proved the link between genetics and molecular structure. It's a classic example of a one-letter mistake in the genetic code having just catastrophic consequences.
Linus Pauling was the first to propose it was a molecular disease.
He was. And the specific defect, discovered by Vernon Ingram in 1956, is remarkably small.
It's a single amino acid substitution in the beta chain.
Glutamate is replaced by valine at position 6, glutaval.
And glutamate is negatively charged, it's hydrophilic, it loves water. Valine is small and entirely nonpolar hydrophobic.
So you switch a friendly, water-loving, charged residue for a water-hating, greasy one right on the surface of the protein. And the consequence of this tiny change is catastrophic.
What happens?
This substitution drastically decreases the solubility of deoxyhemoglobin S, the T state. The new nonpolar valine 6 residue creates a sticky hydrophobic patch on the surface of the molecule.
And that patch fits into a complementary hydrophobic pocket on a neighboring deoxyhemoglobin molecule.
Right, a pocket formed by phenylalanine 85 and leucine 88. And this hydrophobic attraction acts as the initiation point for polymerization.
So the molecules start to stack up.
They stack and aggregate into these massive, rigid, fibrous cables made of 14 separate hemoglobin chains. And these fibers distort the normally flexible red blood cell into a rigid, crescent-like sickle shape,
Leading to the clinical symptoms. Clogged capillaries, painful crises, and anemia from the short-lived red cells.
Exactly.
But why does this only happen when the cells are deoxygenated, like in the tissues?
Because that aggregation site is specific to the T state, the complementary binding pocket, the one formed by Phe-P85 and Lu-88. It's largely buried and inaccessible when the protein is in the oxygenated R state.
So only when oxygen tension is low, forcing the protein to snap into the T state, does that pocket on the surface become exposed.
And able to interact with the Val6 patch on a neighboring molecule. The disease is triggered by the function of the protein, not just its existence.
It's fascinating. Now, the paradox. Why is this dangerous mutation so prevalent in regions with high malaria rates, like West Africa?
This is a textbook example of balancing selection. If you inherit one normal gene and one mutated gene, you have sickle cell trait.
Which is usually benign.
Usually. But having the trait confers significant protection against malaria caused by Plasmodium falciparum.
How does it protect against malaria?
Well, the precise mechanism is complex, but one leading theory is that when the malaria parasite infects a red blood cell, it consumes oxygen, which lowers the internal PO2.
So that forces the HBs inside the cell to polymerize, even in carriers.
And that premature sickling causes the infected cells to be rapidly filtered out and destroyed by the body's immune system before the parasite can fully mature and spread.
So the selection pressure for malaria was so intense that it favored the survival of carriers, despite the risk of passing on 2 copies and causing full sickle cell anemia.
Exactly.
So sickle cell is a problem of protein quality. Other major hemoglobin diseases, the thalassemias, are problems of quantity.
That's a great way to put it. Thalassemias result from the loss or a severe reduction in the production of one of the globin chains. You just don't make enough functional hemoglobin.
Let's start with alpha thalassemia, where you don't make enough alpha chains.
In alpha thalassemia, the excess beta chains that are left over form these unstable tetramers called hemoglobin H, or HbH.
And HbH is a functional failure.
Completely. It binds oxygen with extremely high affinity, but critically, because it's missing those alpha-beta interfaces, it has no cooperativity.
So its binding curve is hyperbolic, just like myoglobins.
Right, and the result is poor oxygen release in the tissues, which leads to hypoxia and severe anemia.
And what about beta thalassemia, the more common and often more severe form?
Here, beta chain production is deficient, and the excess orphaned alpha chains are highly unstable by themselves. They don't form functional tetramers. Instead, they misfold and form these insoluble aggregates that precipitate inside the precursors of red blood cells,
And those precipitates trigger the destruction of the immature red cells.
leading to severe transfusion-dependent anemia, which is known as thalassemia major.
The sources noted that severe alpha thalassemia is less common. Why is the genetic risk different between the two?
It's a matter of gene dosage. We typically have only 2 alleles for the beta chain, 1 on each chromosome 11. But humans usually have 4 alleles for the alpha chain, clustered on chromosome 16.
So to get severe beta thalassemia, you just need to disrupt both of your alleles. But for severe alpha thalassemia, you'd have to disrupt 3 or 4, which is statistically much rarer.
Exactly.
This brings up another protective mechanism we might not think about. Red blood cell precursors normally make a slight excess of alpha chains. If those free alpha chains are so prone to aggregation, how does the body prevent this problem in healthy people?
That is the job of the alpha hemoglobin stabilizing protein, or AHSP.
A chaperone protein.
A chaperone protein. It's a small 11 kiloday protein that forms a soluble complex specifically with newly made single alpha chain monomers. And it binds precisely to the fates where the beta chain would eventually connect.
So AHSP is like a temporary scaffolding.
That's a perfect analogy. It ensures the alpha chain is correctly folded and prevented from aggregating until the beta chain is fully expressed and available. Once the beta chain arrives, it displaces AHSP and you form the stable alpha-beta dimer.
And that's the housekeeping mechanism that fails in beta thalassemia when the beta chain never shows up.
Right, leaving the alpha chains vulnerable to aggregation.
Finally, we should just acknowledge that the globin family is bigger than just alpha, beta, and gamma.
Oh yeah. Our genome contains other related globins. We have 2 key monomeric relatives. Neuroglobin, found mostly in the brain and retina, where it might protect neural tissue from hypoxia.
And cytoglobin.
And cytoglobin, which is more broadly expressed, and its exact cellular role is still being figured out. Structurally, what's unique about them is that in their deoxy form, both the proximal and the distal histidines are coordinated to the iron atom.
So oxygen binding has to physically displace one of the protein tethers before it can settle in.
It's a slightly different mechanism.
We spent a lot of time on the structural and conceptual basis of cooperativity. Now let's get quantitative. We rely on mathematical tools to prove these concepts, starting with the Hill plot.
The Hill plot is over a century old, but it provides a rigorous, visual, and simple way to measure cooperative binding behavior.
So biochemists plot the logarithm of the fractional saturation ratio against the logarithm of the oxygen partial pressure.
That's it. And the most important output from this graph is the slope of that central linear region. That's the Hill coefficient, or N.
And what does that number N truly represent?
The Hill coefficient is an empirical measure of cooperativity. For a non-cooperative protein like myoglobin, N equals 1. If a protein had perfect instantaneous cooperativity across all 4 sites, N would equal 4.
And the observed Hill coefficient for hemoglobin is 2.8.
Right, and that 2.8 isn't just a number, it's the mathematical proof of significant but not absolute cooperativity. It confirms that binding of oxygen doesn't instantaneously shift all 4 sites.
It allows us to calculate how steeply the affinity changes.
And a binding curve calculated using n equals 2.8 perfectly matches the complex physiological sigmoid curve. It proves that this mathematical simplification really captures the essence of the complex allosteric mechanism.
We also talked about the concerted, or MWC, model qualitatively, but we can also model that T to R structural switch mathematically using just a few parameters.
This is the elegance of the MWC model. We can characterize the whole system using only 4 parameters: N, which is 4, the number of binding sites; Kr, the dissociation constant of the high affinity R state; C, which is the ratio of Kr to Kt, describing the affinity difference between the 2 states; and then the essential parameter L.
Let's focus on L.
L is the allosteric constant. It's defined as the ratio of T-state molecules to R-state molecules when 0 ligand is bound.
So it tells us the inherent natural structural preference of the protein in the absence of oxygen.
Exactly. And when biochemists fit the observed physiological data to this equation, the optimized parameters are really revealing.
What are they?
The best fit requires L to be 9,000, C to be 0.014, and Kr to be 2.52.
L equals 9,000. That is a powerful piece of information. It quantitatively proves that in the total absence of oxygen, the tense, low-affinity state is inherently favored over the relaxed, high-affinity state by a staggering ratio of 9,000 to 1.
This huge value confirms why we need 2.3 BPG. Without those internal stabilizing factors, the protein would default to the high-affinity R state way too easily. The L equals 9,000 ratio is the energetic foundation that makes the T state the primary default.
And C equals 0.014 tells us that the R state binds oxygen roughly 70 times more tightly than the T state.
Exactly. The low C value and the high L value work together to generate that perfect sigmoid curve. And the quantitative switch is dramatic. The model shows the population ratio flips from 9,000 to 1 with T favored at 0 oxygen bound, all the way down to 0.0000035 to 1 with R favored when all 4 oxygen molecules are bound.
So this modeling provides the elegant mathematical proof that the mechanism driving efficient oxygen transport really is the population switching almost completely from the T state to the R state upon oxygen loading. It brings the structural insight into the realm of precise numbers.
It does. We've really completed a comprehensive tour of hemoglobin, moving from its evolutionary roots all the way to its molecular mechanics and mathematical description. The key takeaways, I think, confirm its status as a masterpiece of biological engineering.
First, hemoglobin's efficiency, delivering that massive 66% of its capacity, is achieved entirely through its quaternary structure, which enables the hallmark of cooperative binding and that necessary sigmoid curve.
Second, that efficiency is driven by a precise structural mechanism. The 0.4 angstrom shift of the iron atom acts as the mechanical trigger, transmitting strain through the proximal histidine and alpha helices, driving the tense-to-relaxed transition across the entire tetramer.
And third, the system is exquisitely regulated by allosteric effectors. Internal stabilizers like 253 BPG enforce the low-affinity T state, while external metabolic cues, hydrogen ions and carbon dioxide, activate the Bohr effect, guaranteeing oxygen release precisely in the most active, demanding tissues.
And finally, we learned that a single amino acid substitution, the glutathione change in hemoglobin S, reveals just how delicate the structural balance is. That change creates a fatal surface aggregation site that is only exposed in the deoxy, or T state, resulting in a devastating yet evolutionarily preserved disease.
It is truly stunning how fundamental principles of chemistry and mechanics dictate life at the physiological level. And here's a final provocative thought to leave you with, building on the brilliance of the Bohr effect. Consider the molecular elegance of how the cell's waste products H+ and CO2 are leveraged to become the precise chemical signals that trigger the release of its most vital resource, O2. It's like a perfect self-regulating feedback loop at the protein level.
It really is.
So it makes you wonder, what other fundamental biological processes might be regulated by molecules we currently dismiss as just, mere byproducts of metabolism?
That's something powerful to contemplate. Thank you for diving deep with us today.